The next person on this wandering through careers that are or are not directed at a single goal. Charles Cantor from Sequenom. Is that the way you pronounce that? Sequenom. So the accent is on the last syllable. Non-invasive personal genomics. Actually, I'm not sure how I feel about that.
Actually, I think I was invited to talk here because Larry and I spoke at the same symposium in Japan about a year ago, and he was very envious of this title. But in view of the two previous talks, I think I'm going to rename this and speak about how to do small science on a big budget. Now, it's small science because it's diagnostics, and because, as you'll see, we're going to deal with painfully small numbers of molecules, almost really the limit of what's possible. And it's a big budget because what we're doing is, unfortunately, quite expensive. So it's been 10 years since we've had one genome sequence for humans, and now we have something like 1,000, and tens of billions of dollars have been spent. And it's fair to ask, has this changed the practice of medicine? And the answer, unfortunately, is not very much, except in 3 areas. One of those is age-related macular degeneration, which I'm not going to talk about today, because that's a complex disease which turned out to be very simple. The other 2 are prenatal diagnostics and cancer. And the reason why knowledge of the human genome has changed those 2 areas fundamentally is because the genetic effects, the DNA effects, are very penetrant. They're very strong, and they're simple in those 2 areas. And in most other areas, it's very gray. So we look at DNA because it's predictive of phenotype. It's predictive of properties. And in this talk, I'm going to make what may seem like a far-fetched analogy between 2 very different clinical situations, which are also in very different stages of development. What's common between cancer and pregnancy is that in both cases you have a foreign object growing in the body of an otherwise normal host. In one case it's the tumor, in the other case it's the embryo. And what's of interest clinically is looking at DNA sequence differences between the foreign object and the host. And there's a long history of doing this by invasive procedures. You take a tumor biopsy, or you do something like amniocentesis, and you actually get a sample of the fetus. And you look at DNA sequence information and draw conclusions about the properties of the fetus or the characteristics of the tumor. But those procedures are invasive. You can do them once, but most patients are not going to tolerate you're doing them many times. What I'm going to show you is the progress in transforming those invasive procedures into non-invasive procedures, which cause less patient discomfort and also, if necessary, can be repeated. So in the peripheral circulation, there is DNA, of course, inside nucleated cells. But in addition, there's a very tiny amount of DNA that is not in cells. And we call this cell-free plasma DNA because, as this slide indicates, we're not exactly sure of what state it's in, except we know it's not in cells. And the cell-free DNA actually represents only 1 part in 100,000 of the total DNA. And so all early attempts to study it were tremendously compromised because people broke significant numbers of nucleated cells and contaminated this cell-free DNA. But we know how to avoid that cell breakage. So the origin of this free DNA is apoptosis, which means programmed cell death. So we are seeing images of dead cells. It's very short, so it was missed by most methods of DNA purification. It turns over very quickly. It has a half-life of only 15 minutes. So it truly is a snapshot. And we don't actually understand how it's packaged. So we're going to look at two aspects of this free DNA. Its sequence and its quantitative representation to try to draw biological inferences. And if I just focused on the simplest possible genetic situation I could have, what we call a single nucleotide polymorphism, if I look at just one sequence position in the human genome, and since humans are diploid, and if it's a biallelic locus, it has two possible sequences, then the situation's very simple. I have these three possible genotypes, which you all learned about in high school or maybe even elementary school now. And if I focus on the abundance of, let's say, the small a sequence, then it has three possible values. But things get much more complicated because in the human genome, many genes exist in multiple copies. And so just with 2 copies alone, I have 5 possible genotypes. And if I have many copies, I have many possible genotypes. And these are all samples that are pure. And so allele frequencies are discrete valued functions. They're discontinuous. But if I look at mixed samples, such as fetal DNA or tumor DNA in the blood, or tumor DNA in a biopsy, since those samples are mixtures of different types of cells or different types of origin of DNA, then these allele frequencies become continuously variable functions. So you can see already that to draw inferences, I'm going to need to have very high sensitivity to see very small abundance. And I'm going to have quantitative precision. But the thing that really makes this brutal is a phenomenon called stochastic sampling noise. Because we have this wonderful tool, the polymerase chain reaction, we can start with a single molecule and amplify it to the point where we can determine its properties in the test tube. But that doesn't mean that we can do this reliably, because the problem is statistical fluctuations. If on average I have 1,000 molecules in a sample, but it's a sample, then my noise is about 30 molecules, and my detection threshold is about 3 molecules, because a significant fraction of the time, there won't be any molecule at all of a particular sequence. And I'm going to show you cases where we're dealing with 100 molecules, or perhaps even 10 molecules. And what that means is that it is fundamentally impossible to make quantitative measurements on such small numbers of molecules. I can detect things, but I can't quantify things. Okay. But we're interested in quantifying things. We want to know whether a baby has the right number of chromosomes. And that's a quantitative measurement. It's not a sequencing measurement. So how do we overcome this problem in fundamental physics? There are 2 ways. The obvious way is to repeat the measurement independently. And if I do 100 replicates, I can decrease sampling noise by a factor of 10. And if I do more replicates, I can decrease the sampling noise more. The problem is there are so few molecules that to make a single measurement, I need a cc of blood. In fact, I need 10 cc's of blood. Okay? So I can't repeat the measurements 100 times, because that would require a liter of blood, and that's just not practical. Okay? So this doesn't work. The alternative is to use genetics cleverly and realize that in the human genome, although I have very few molecules that derive from each location in the genome when I look in plasma, I have many locations in the genome that tell me the same answer. For example, if I'm interested just in counting chromosomes, any sequence that I can assign to the same chromosome is equivalent. Or if I'm doing more fancy things, for the geneticists in the audience, I can use haplotypes to use lots of equivalent sequences that are relatively nearby. And so by averaging the results, not from 100 samples, but from 100 equivalent loci, I can reduce the noise. But you'll see, we really are treading on the edge here of what's possible statistically. Okay. So non-invasive prenatal diagnostics for the common aneuploidies, trisomy 21, 13, and 18, became a commercial reality in the US in October of 2011. And just at Sequenom alone, we anticipate this year testing 150,000 high-risk pregnant women. To put that into context for you, there are 750,000 high-risk pregnancies in the US. So we will have captured, basically 18 months after launching a product, 20% of the market. And there are 3 other companies competing with us. So people have said this is the fastest adoption of a new medical procedure since penicillin. And it's probably safe to say that within a few years, most pregnant women will be tested in this manner that I'm going to show you. But right now, it's only available for high-risk women, women over the age of 35, or other indications of high risk. The procedure is to draw 10 Cs of peripheral blood from the mother's arm, isolate plasma, and as you'll see, randomly sequence the free DNA molecules in plasma to assemble a crude map of the human genome. And we use the entire human genome sequence, but we don't measure it in that detail on each patient. Okay, now in an average pregnancy, it turns out that 13% of the free DNA in the mother's blood comes from the fetus, and the remainder, of course, comes from the mother. This is independent of gestational age. And so, no, we really don't understand where this number comes from, okay? The fetus is obviously much smaller than the mother, but most of the fetal DNA comes from the fetal portion of the placenta, which, like a cancer, has a lot of cell death, okay? The problem is that in different mothers, the level of fetal DNA varies by about a factor of 20. And nobody knows why. So imagine a situation where a mother has so little fetal DNA that there aren't enough molecules to detect. Then we will conclude that the fetus is female because the mother is female, and we'll conclude that the fetus is normal because the mother is at least normal enough to be pregnant. And so this will be a false negative if the fetus is otherwise, okay? So we have to have a way of qualifying each clinical sample to be sure that there is enough fetal DNA there to avoid a false negative. Now, if the fetus is male, this is trivial because the mother has no Y chromosome. So if we look for sequences that are assigned to the Y chromosome, they are uniquely fetal. But half of all fetuses, roughly, are female, and they have no Y chromosome. So we have to do this another way, and we take advantage of a phenomenon called imprinting, or epigenetics. The mother is developed, the fetus is developing. There are certain genes that a developing fetus must not express. And so those genes become methylated to shut down those genes in the fetus. And we search for sites which are methylated, shut down in the fetus, unmethylated in the mother, and which fall in nucleic acid sequences which are recognized by enzymes that can cut and destroy the unmethylated maternal DNA, but can't touch the methylated fetal DNA. So basically, starting with a mixture of mother and fetus, we destroy the maternal sequences. What's left is pure fetal, and we just count it. Okay? It's a very useful trick. So here's what one of those regions look like. We use many to get statistics. In women who are not pregnant, this is the number of molecules per cc of plasma. In women who are not pregnant, there are no methylated molecules. Even earlier than 8 weeks, we have a robust signal. And 8 weeks or after, when we would actually do prenatal diagnostics, we have a strong signal. But you can see the variance from mother to mother is tremendous, and the mean is 140 molecules. So that's what we're dealing with in an average case. And in a bad case, we may be dealing with 20 molecules. Okay, well, the most common aneuploidy is trisomy 21, but 13 and 18 are also significant. Okay, so here's a cartoon of what I've said so far, that in an average pregnancy, a minority of the DNA fragments come from the fetus, a majority come from the mother. The accuracy of current sequencing is that it's hard to distinguish these two. But this is the experiment that we do. It's honestly a mindlessly simple experiment. It's just very expensive. We randomly sequence fragments of DNA from the mother's blood, and where they match a genome assembly, we assign those sequences to the chromosome of origin, and we currently do this 8 million times. And that allows us to build up a representation of the relative abundance of each of the chromosomes in the mother's blood. Some of these counts have been contributed by the mother, a minority by the fetus, but we're not distinguishing. We're just counting. If the fetus has an extra copy of chromosome 21, then incrementally, those counts will be somewhat overrepresented compared to normal. And that's our whole effect. That's the only information we use. Except that we have to use some rather elaborate statistics to make sure that this is a meaningful result. I'm not going to show you those statistics, and you're happy that I'm not going to show you those statistics. But here are the results from a clinical trial that was run by Brown University for us with 27 sites worldwide. And what I'm plotting is the overrepresentation, some particular statistical measure, of counts that correspond to each chromosome as a function of the fraction of fetal DNA that that particular mother had. And you can see that the strength of our signal more or less increases linearly with the fraction of fetal DNA. And what you can't see for sure on this slide, but I'll show you numbers shortly, is that we have a very robust signal. With 8 million sequence reads, we very rarely make a mistake. So from that initial clinical trial, which was published last year, you can see that we're not perfect in detecting trisomies, but we're nearly perfect. So this is as good as a clinical test gets in a research laboratory. But what then was done was to take this research result and transform it into a clinical test with better procedures and more powerful statistics. And so today, roughly the error rate is about 1 in 1,000. And that is comparable to, perhaps even better than the invasive tests. Okay? And to be honest, it's almost arbitrarily good in terms of rejecting false negatives. Because the more we sequence, the better we can make the results. But I can't say the same for the false positives. Because biology is interesting. And the false positives, as I'm about to show you, point to very interesting biology. So first, let me just show you, this is data on 100,000 clinical samples. Once again, here just for chromosome 21, showing you the signal we have, and of course, tremendous, most of these, 98% of these are normal, okay? And so this is really quite good. But we do make occasional mistakes. Okay. If there are any OBGYNs in the audience, the question that I always get asked, but now I'm anticipating it and answering, is a phenomenon called confined placental mosaicism. There are pregnancies in which the fetus is normal, but the placenta is down. And most of the DNA that we see comes from the placenta. So those will be false positives because it'll look in the blood like it's trisomic, but it's not. 3.7% of all our pregnancies are twins or higher multiplets. And it's common, especially in vitro fertilization, that you lose some of the implanted embryos, but after the placenta has formed from those embryos, okay? So now you have a situation called a vanishing or a lost twin, in which we think the twin is still there because we see DNA that's coming from the placenta. And if the twin that died was aneuploid, and most aneuploid twins die, then we make a mistake. And there are other things that we can see. The fetus can be a mosaic and not a uniform chromosome composition, or the mother can, or the mother has cancer. So here's a case in which there's too much chromosome 21, there's too little chromosome 13, there's too little chromosome 18. No fetus with this genome can survive, but of course tumors are less demanding, and in fact this mother had metastatic carcinoma. Here's another case, and we repeated this result just to show you how good our methods are, and once again this mother had cancer, okay? So the methods that I'm showing you, while developed for prenatal diagnostics, exactly the same methods are applicable in cancer. But as you'll see, it's more complicated in cancer. Okay, now the problem is that in addition to the aneuploidies we're looking for, which we understand clinically and have strong predictive value for the patient, we see other things. Here's a patient, pregnant woman. We're looking across the genome. Here's chromosome 18. It's not present in 3 copies, but there's too much of part of it. Unless this is a well-known so-called genetic syndrome, when we see this, we don't know if this is the mother or the fetus. We just know something's wrong, okay? Now we can decide between the mother and the fetus by using maternal nucleated cells to look at the mother separately, okay? But what I'm telling you, alas, because we're using sequencing, which is really a discovery tool, we're using it in a clinical testing paradigm, we see things that you don't want to see, okay? Here's a bunch more. We see these in 7 cases out of 1,000, okay? Here's a chromosome 1p36, a deletion, a duplication, and so on. I bet every one of you has at least 1 million base pairs missing or extra on one chromosome. Obviously, it hasn't affected your state of health or quality of life too much, but we all have these things. Okay, so the better we get, the more deeply we sequence to try to avoid making mistakes, the more we see these things. So about 2.5 years ago with Dennis Lowe, we sequenced the entire genome of an unborn fetus. That single experiment cost us $250,000 in supplies. This year, 2 groups repeated it with slight variations in procedure, and they were able to bring the cost down to $50,000 in supplies. And just extrapolating in 4 or 8 or 10 years, this will become an affordable test in every pregnancy. And it raises ethical issues which are mind-boggling. And I could spend the rest of the talk on them, but I'm not going to. Suffice it to say, we better get prepared to deal with this kind of information. Instead, I want to use the remainder of my time to switch to cancer. It's a lot less mature field. It's not accepted clinical diagnostics. It's still a research tool. But in the last year, there's been a flurry of publications, some by our customers, some by other independent groups, showing that the same sorts of things that I showed you in pregnancy are going to be possible in cancer. The difference, and it's very important to understand this, is that when we're looking at a fetus, it's a stable genome. It's not changing. One measurement suffices. We know the parents, in principle. Well, you heard the comment this morning. It's actually 10% mispaternity in the US is our number, much higher in certain countries. So we know the parents in principle, and we can use Mendelian genetics, which is what we did in cases like this, to simplify the untangling of which fragments come from mom and which fragments come from the fetus. In cancer, the genome is unstable. It's basically going to hell. And so we don't have those powers. So this is the first publication that I'm aware of that looked in a clinical cohort of significant size, looked at tumor-derived DNA, free DNA once again, in plasma using modern methods which are not confounded by prior complications. And this comes from the group of Johan de Bono in London. These are phase 1 clinical trial patients, which means they have late-stage fatal disease. And probably, although they're being treated, the treatment is not effective. So they're essentially untreated at this point. All right, now the bottom 2 rows are de Bono's measurements of the amount of DNA, free DNA, from all sources, patient and tumor, in blood. And that's our average number in pregnancy. And I don't think the difference here is significant because 2 different methods were used to make this. This is total free DNA. But obviously, since the same method was used for the healthy volunteers in London and the late-stage cancer patients, this factor of 7 increase in plasma DNA on average is very significant. Now, it is tempting to draw the conclusion that most of this DNA is from the tumor. Okay. This doesn't prove it, and nothing I'm going to show you in this talk proves it because of constraints of time. But I can prove it to you that much of this, by the same kind of epigenetic tricks that we use for pregnancy, that most of this DNA is actually coming from the tumor. Now, once again, what makes this extremely difficult for us and for de Bono is that the patient-to-patient variation is enormous. So this is colorectal cancer, breast, et cetera, healthy volunteers, and we're looking at the just the total concentration of free cancer DNA in plasma, and you can see once again it's about a factor of 20 broad, and we don't know why. So the first question that you like to know is, can I see in plasma DNA sequences that are particular to the tumor and that assist in making therapeutic choices? And Tim Harris, of course, is in the audience and highly inspired by a great review article you wrote a few years ago. The answer is yes, you can see this, but it's not simple. I'm going to spare you a very complicated slide. At the current time, the concordance between what you see in an actual physical sample of the tumor, a biopsy, and what you see in plasma is about 50%. And that's a combination of two things. The methods being used by most people are not sensitive enough to see below a threshold of about 1% in plasma. And two, this is much more interesting. Remember that what we see in plasma is DNA from cells that have died. But what you see in the sample of the tumor is DNA from cells that are predominantly alive. Okay? Though there's no reason to think that the plasma will necessarily represent the properties of the tumor. That's a subject for future research. What is clear is that just the total concentration of free DNA in the plasma of a cancer patient has tremendous prognostic value. So patients with very little free DNA in their blood have, in this case, we're looking at actual survival because these are phase 1 clinical trials. They have very good prognosis. Patients at the very high edge of plasma DNA have very poor prognosis. And there's no reason why you should believe a result like this from a single clinical site. But in the last few months, an exact replica has come from a second clinical site, and so now we really have to take this seriously. Sadly, this very useful measurement could have been made 50 years ago. They're just measuring concentration of free DNA in plasma. No sequencing, no nothing, okay? Except that nobody knew how to prepare the samples properly. Okay, but here's one example of how this is therapeutically useful. Patients with lung cancer often have activating mutations in a gene called EGFR. That renders them susceptible to several personalized cancer medications that we now have available. But unfortunately, after treatment or in rare cases even before treatment, these patients have a mutation in the target which makes the drug less effective, and this is called the T790M mutation. And so a group in Japan looked in plasma and was able to show that patients who had this T790M mutation in red had a much worse prognosis. In this case, it's progression-free survival. So they had about twice as fast a progression as patients without. So this kind of approach is genuinely useful, and honestly, we're at the tip of the iceberg here. Okay. Now, we're able, using tricks, to see mutations in plasma at very low levels. The tricks are technical, but basically, we can focus in just on variant sequences, and as the next slide shows, these are spike-in synthetic samples. We can see 1% mutants, we can see 0.25% mutants. We have no background. Okay? So the good news is we have exquisite sensitivity. The bad news is we're in the range where we can't quantify, okay, so that we can see these things, but we can't quantify. So what I want to do is spend the last 2 minutes, since the theme of this conference is something like sociology of medicine or societal changes of medicine. I forget now what the lovely title was that Larry made up. So let me tell you just an anecdote and then make a pitch. In Japan, until last month, there was no prenatal testing whatsoever. The Japanese did not do any serum screening. They did not do routine ultrasound. They did not do invasive prenatal testing, except under the rarest of circumstances, because abortion in Japan is illegal. And so starting only 18 months ago, we made a pitch to Japan that they should adopt non-invasive prenatal testing because it was risk-free, it was very accurate, and Japan, like most westernized countries, developed countries, I should say, is having increased maternal age. So 28% of all pregnancies in Japan are over age of 35, right, for the mother. So, I mean, they're having an epidemic of Down syndrome. And against everybody's guesses, 16 months after this campaign was initiated, the Japanese government approved prenatal testing in Japan, still in a limited, for a limited set of tertiary care hospitals with good genetic counseling. So it is possible to change the way a country views the practice of medicine. Okay, so the pitch. What I showed you in pregnancy is straightforward because you only have to make the test once for each pregnancy. What you'd really like to do in cancer is routinely monitor patients before they get visible evidence of the disease for early detection, and then throughout all phases of therapy, remission, and so on. And of course, with the non-invasive test, you can imagine doing this. But the problem is the interpatient variability. So probably the only control for each patient is the patient himself. And so what's needed is a baseline study. What's needed is for someone to make the very substantial investment of taking a large cohort of people who are healthy and following them yearly or more frequently by plasma DNA measurements and sequencing until they get sick, and then going back and looking at the data and having the right markers. And of course, you could do this with nucleic acids, with proteins, or so on. But we're talking very big bucks here. And it's going to take great courage on somebody's part, whether it's private sector or public sector, to make the kind of investment that would be needed to do this. And it will take 20 years to get the results. But if we don't start this year, it's just one more year before we have these benefits. Thank you.
I am no great friend of molecular genetics, thinking that we're chest deep in unfulfillable promises. But this is one that just rouses great excitement, in particular because it appears to me that this is the key to distinguishing between those people who have a cancer. If a pathologist gets to cut up an organ like the prostate at post-mortem and finds one or more loci that they would say this is malignant, and the person died of something else, and to be able to have some countervalence against the culture of medical activism. Do something. Don't just stand there, do something. That's a comment more than a question, but I wonder if you see that potential.
Well, the way this is going to start, almost certainly, is in monitoring treatment. And in particular, monitoring treatment with these very expensive personalized medications, which $5,000 or $10,000 a month. Because by monitoring the mutations in plasma, you will know within a month or two whether the patient is responding or whether the tumor is just changing its properties and, escaping. And so there will be, we're always told in medicine these days, if you don't reduce the cost, you're not going anywhere. So it looks like these tools will help avoid wasting money on drugs which are not going to work for a given patient. But honestly, this is very early days in cancer. Cancer is much more complicated than pregnancy.
Charles, over here, Charles.
Yes.
Will your tests work on frozen blood samples? Because if they would, longitudinal cohorts like the Nurses' Health Study, which has been going on for 30 years, would be the kind of baseline samples, longitudinal, with outcomes that would help you.
Okay, so the answer makes me cry because it will work on frozen samples if they were treated in certain ways, which we know now, but they didn't know then. So I would like to get my hands on that Norwegian nurses study, which is a very famous study. I'd like to get my hands on some of those samples, which are closely guarded, to see whether any of them were properly collected. And there are similar cohorts on, longitudinal cohorts on Alzheimer's, which would be fantastic because almost certainly in Alzheimer's, brain-specific DNA will be found in the circulation. But once again, you're faced with a 20-year time frame. And I am just afraid those samples are compromised too. So the challenge, and when I think about the baseline study I want to do, is how do I guarantee to anybody that the methods I have now will be adequate for the diagnostic test I want to develop 20 years from now? And of course, I can't guarantee that. But yes, frozen cells work. But even better is a trick, a new special blood collection tube, which made by a company called Streck, which doesn't break the cells. It's these little things that make all the difference, right?
Well, that was basically my question, because it would reduce the cost a lot of your prospective study if you didn't have to sequence everyone until you actually knew. So what would be the indication that you'd like to start now by just freezing the cells and following people until there was an event that you could then go back and sequence those frozen cells?
Well, I would prefer to do this across all indications. Just bite the bullet and do it right. That's what was done with the Norwegian nurses study, right? There was at least 20,000 nurses, right? Yeah. That's what I'd like to do. This requires a big donor.
Two quick questions. One is, have you seen trisomy of any other chromosomes?
Okay, let me answer that. Of course, we see trisomy of any other other chromosomes. Okay, trisomy 16 is the fourth most common. But now I have to tell you, we don't report. Okay? The reason why we don't report, there are two reasons why we don't report. Those other trisomies inevitably spontaneously abort. Inevitably.
Yeah.
Okay? We see a hell of a lot of 18. Most of those will not go to term. Okay?
But you do report
But the other
those.
problem we have is for the rarer trisomy, I mean, we've now studied 100,000 women. We have no statistics on those rarer trisomies, something that happens 1 in 10,000 patients. We don't have confidence limits on the results. And so we can't really honestly report them.
The second question is, have you seen essentially mosaicism of T21?
Yes, yeah, we have no trouble getting most of the mosaics right. Okay, and nobody asked, we have no trouble getting most of the twins right, too, right?
I have one right here. My question is more sociological. You had mentioned, again, this is on the prenatal testing off of Tom Blumenthal's question, that it's possible to change how a country uses prenatal testing or medicine when it came to Japan, and that was a big progress. My personal experience was when I had an amnio, I was told my baby with Down syndrome would die by the time she was 3. And then I had to make a decision whether to abort or not. And I didn't know what decision I would make. So is it possible, and is it your responsibility as Sequenom to ensure a country or a society's medical profession, the professionals, administer these tests in a non-coercive way, providing accurate information? How do you deal with the science catching up with the societal issues?
Well, I can tell you the solution in Japan, which was straightforward, and that the test is only offered after appropriate genetic counseling, and then followed up by appropriate genetic counseling. The reason why Japan has restricted it at the moment to a set of tertiary care hospitals is because only they have the genetic counseling. In the US, most of the people that we offer the test to are large maternal fetal specialist clinics that have genetic counseling, and at Sequenom we back them up with genetic counseling as needed. Unfortunately, in your personal case, where you were told was simply wrong, and that happens, right? Yeah.
Charles, let's say it takes 7 mutations to get to colorectal carcinoma, and the person with 6 has a benign polyp, and the person with 7 has an early stage colorectal cancer. Are you suggesting that taking blood periodically from this person, this is going to be a reliable technique to determine whether a person has a benign polyp or an early stage colorectal cancer?
Right. Okay. So if the number of driver mutations was as large as you say, no. But it isn't. Okay? It's generally 2 or 3. And I mean, again, it's very early days, but it's generally 2 or 3. And in some cancers, I actually skipped a few slides, but in some cancers, we understand the mutation spectrum very, very well. Lung cancer, colon cancer, we basically can provide rational explanations for something like 70% of patients. Other cancers, prostate, ovarian, it's terrible. We know nothing. But so it's cancer by cancer, patient by patient, yeah. But some of this is clear-cut. Unfortunately, some of it is not.
I understand that the triplication of 21 is not the same triplication in each patient. So presumably you've got statistics to analyze just what bits are triplicated and what bits are not. Because if I recall correctly, alpha-syn is on that same particular piece of DNA. And I'd be interested to know how often alpha-syn is triplicated and how often it isn't.
Right. So Tim, that's a very complicated question, and I'll answer it only in part given the time, okay? In 90% of Downs, the extra chromosome comes from the mother. In 10% it comes from the father. So that's the first variation. We don't today distinguish that, okay? In a certain fraction of Downs, the extra copy comes, sorry for the technical term, from what's called a Robertsonian translocation, okay, which you understand. And in that case, of course, the size of the triplicated piece varies, or can vary a lot. And our sensitivity in an average pregnancy is probably about 1/6 of a copy of chromosome 21. Okay? So whether it's a mosaic situation and 1/6 of the cells have the whole extra 21, or whether it's a translocation or some other weirdness, and the patient has all the cells are the same, but they have only a 1/6 copy of chromosome 21, then we will see that. But your question is more penetrating, because as you know, it's only part of chromosome 21 that introduces the phenotype. And yes, we would know, as in the few cases I showed where we sequence somewhat more deeply. If we wanted to, and in those cases we probably would go back and sequence more deeply, we would know whether the fetus has gotten what's called the Downs critical region of chromosome 21, or whether they've got extra parts of chromosome 21 that nobody cares about. But
Okay.
this will develop as, sequencing becomes cheaper. We look in more detail. Yeah.
Thank you very much.